Hey, all, Miss O'Leary here. I am going to walk you through the lac operon processes. This is very similar to how I would do this drawing out on the board in class, so hopefully it will be helpful for you. So first of all, these are the parts of the operon. When we talk about the lac operon, we are often referring to the whole process of regulation related to lactose metabolism, but specifically the operon is the part to the right here with the POZYA. And these other parts are regulatory. But there are some things that are familiar to you, hopefully, from looking at gene expression. The promoter is noted with a P and the terminator with a T. And note that there is a promoter for this gene here, and then there's one for this whole operon over on the right. In terms of what these genes actually are, starting on the left here, this is the CAP gene. The CAP protein that's made from this gene is an activator. The I is the gene to make the inhibitor protein. And then here we have the CAP binding site. It doesn't have kind of a cool short name, but this is where the CAP protein would bind. Again, this is our promoter for the operon. O is the operator, which is the inhibitor binding site. Z is the gene that codes for beta-gal, the beta-gal protein, which you should be familiar with. Y is the gene that codes for permease, which creates a doorway for lactose entry. And then the last gene in the operon is a transacetylase, here noted with an A, but we aren't really going to talk about this because it's not part of the regulatory system. We will talk about the functions of these genes and everything as we go through. So let's start with the regulatory proteins being made over here on the left. So first, they are transcribed just as you would imagine, as we talked about in gene expression. So in the region between the promoter and the terminator into RNA. Remember that this is a prokaryote that we're talking about, so there are no modifications or anything that has to happen after transcription. And then they are translated into their proteins. Of note here, the inhibitor protein is immediately active. So in its three-dimensional shape, after it goes through protein folding, it is functional. However, the CAP protein over here on the left is not functional in its original form. So it is inactive when it is first made. These proteins are made what we consider constitutively. So that means that they're produced all the time. So the presence of lactose or glucose or anything like that has no impact on the actual production of these proteins. Their function is regulated in other ways. So just remember, there are always CAP proteins and inhibitor proteins in the cell. Okay, so now we're going to talk about how lactose helps regulate the expression of the operon. So in the first place here, we have our inhibitor proteins that are being made. And in the absence of lactose, the inhibitor proteins, like these horseshoe shapes, will bind to their binding site, which is the operator. When the operator has the repressor protein on it, that means that the RNA polymerase cannot bind to the promoter. So even though the RNA polymerase actually touches the DNA at the promoter, it's a pretty big protein. And so if the inhibitor is here, there's basically not enough room for it to bind and start transcription. So if the repressor is there, the RNA polymerase does not bind. And if the polymerase doesn't bind, that means that there's no transcription and no translation of the operon. But sometimes the inhibitor comes off of the operator. This is not because of any change in condition. This just has to do with reversible binding. So it is bound, and then at a certain rate, it will unbind from its binding site. So think about how this relates back to a power switch versus volume control when we talk about gene regulation. So a power switch would be a repressor or an inhibitor that is permanently bound to the operon. And that would mean that that operon could never be transcribed and those proteins never made. However, when you have volume control, think that even when you turn the speaker all the way down, there's still power going through it. So that's essentially what's happening here. You have this reversible binding, which means there's a little bit of stuff happening. And so when the inhibitor is temporarily unbound from the operator, that means that the RNA polymerase can bind and sneak through and make a few transcripts so that you get this low level of expression. Sometimes we will note the reversible binding, kind of like a chemical equation up here on the right here. So the inhibitor plus the operator makes this inhibitor-operator complex, but at a certain rate, it is reversible. So another way that the inhibitor can be removed from the operator to allow transcription of the operon is by lactose, and this is a more active removal. So when there is lactose in the cell, that's the lactose is these little red triangles here, lactose will bind to the inhibitor and change its shape. You can see instead of a horseshoe shape down here, it looks like little wings off of the lactose. And so what this means is that the inhibitor can no longer bind to the operator. And if that's the case, then RNA polymerase can bind and make all sorts of transcripts of this operon. And so one thing to remember, because it is an operon, you get one transcript, so there's one RNA, even though we end up making 3 different proteins. Another thing to note here is that the operator is transcribed, so it is part of the RNA transcript. The way that these are separated out is by start and stop codons. So even though you have one RNA, you can still make 3 different proteins. So again, you can see that the operator region does come down here. Think of what this would be in the RNA. So this would be kind of like your 5' UTR. The ribosome binding site would be in this region. And then these are the regions for the protein coding. So Z, this gene has the directions for the beta-gal protein here in blue. Y has the directions for the permease protein. I drew this as a little door because that's part of what its job is to make a door for lactose. And then A is that transacetylase that we're not really going to talk about. So I put it as an X here. So beta-gal, if you recall, its job is to break down lactose. And lactose breaks down into glucose and galactose. So its main job is to start the metabolism process by cutting lactose apart. Permease, like I mentioned, is a door for lactose to enter the cell. It's important to realize that lactose cannot diffuse through the membrane. So you can't just have lactose, these little triangles are not going to just go through the membrane, they have to actually enter through the door. And so permease is a specific door for lactose. And so it responds to the presence of lactose on the outside and lets it into the inside, where it can change the shape of the repressors and so forth. So you can see as it comes in, it changes the shape of the repressors, both the ones that are free in the cytoplasm, and it will also bind to the ones that are bound to the DNA. And so when the inhibitors are prevented from binding to the operator, or the operator, that allows for increased transcription of RNA. And more transcription means more translation of these proteins. So the cycle continues. We look back one here. So this cycle will continue where you get lactose in, lactose prevents the repressor binding, more transcription, and that means more doors, which means more lactose can come in and make more transcripts and so forth. But remember that beta-gal is also breaking down lactose at the same time. So this cycle will basically continue on until the lactose is gone. So one thing that comes up here is the question of if you need permease in the membrane for lactose to enter the cell, but you need the lactose to be in the cell in order to transcribe for permease, how does the first lactose get in? And now this gets back to our reversible binding here. So if we're back at this picture, even when there's no lactose in the cell, sometimes the repressor comes off. And that means that polymerase can transcribe these genes, which means it can make these proteins, which means there is always a door in the cell. So even when there's no lactose, there are doors. So there's enough permease in the membrane that the first lactose, if it gets introduced in the environment, can come in and start the cycle. So that reversible binding is what allows for the cell to change its function. Okay, so now we have a second sugar, glucose, that will actually help regulate the operon as well. So glucose and lactose are the main sugars that we're looking at. The glucose is the preferred food source of the bacteria over lactose. And when I say preferred, I don't mean like it just likes it. It means that it will preferentially go through the process of breaking down glucose instead of lactose, if it has the option. That should make sense because lactose breaks down into glucose and galactose. And you should know all about metabolism of glucose and how important it is for ATP production. So if the cell can skip a step and go straight to the glucose metabolism, it makes sense for it to do that and save some materials and energy and all that kind of good stuff. Okay, so the glucose regulation has to do with the cap or the activator. So you see here, we still have our inactive cap that's made all the time. But in this case, it's not activated. So in its original shape, it will not bind to its binding site, which means it won't do any activation. In order for a cap to bind to its binding site, it has to have its DNA binding domain open. So this is the place where the protein has affinity for the sequence at the binding site. And the way that the binding domain is open is by another molecule, cyclic AMP or CAMP, binding to the cap protein. So when CAMP, shown here, binds to cap, it changes its shape so that now it can go up and bind to the cap binding site. And if the cap binding site has this protein here, it means that the polymerase will bind more often to the promoter and make more transcripts. So where does CAMP come from? So this, we have to work a little bit backwards here. So CAMP comes from this enzyme over here called adenylyl cyclase, or you can call it AC for short. Adenylyl cyclase converts ATP, which you should recognize, adenosine triphosphate, to cyclic AMP. So the M here is mono, so as in T was in tri, M is for mono. So you have an adenosine and one phosphate. And because of the way the bonds form, it looks kind of like a circle. And so this is why it's called cyclic AMP. And so once adenylyl cyclase does this, then the cyclic AMP can go bind, change the shape of CAP, and then CAP can bind and enhance the gene. So the flow here is that adenylyl cyclase helps to create CAMP, cyclic AMP. Cyclic AMP binds and helps activate CAP. And then CAP activates the transcription of the operon. So rather than just a regular level of transcription, it will happen more often than it would without this activator. Okay, so where does the glucose come in? So glucose I have shown here is these boxes with red inside of them. If glucose is in the environment outside the cell, it can go into the cell through a glucose transporter. This transporter is very similar to permease, but it's specific to glucose. The way that this transporter works, this protein, when it is active, it will inhibit or stop the function of adenylyl cyclase. And so if adenylyl cyclase can't function, then we're stopping the next parts of the process. So that means no cyclic AMP, which means no CAP binding to the CAP binding site. And so again, just to reiterate here, glucose binding to the, or entering the cell will cause AC or adenylyl cyclase to be inhibited. Adenylyl cyclase then cannot create CAMP, which means CAP is not activated, and so no activation of the operon. That doesn't mean that it can't be transcribed, just that the transcription is not enhanced by an activator. So when we look back at our little flow chart here, glucose in the environment inhibits all of this. It is important to note that it is the glucose in the environment that matters here, because we know that when we break lactose down, we do get some glucose, but it is the glucose entering the cell from the outside that causes the regulation of the CAP protein. Okay, so now that you hopefully understand how these two regulators work, our inhibitor protein and our CAP protein, you should think about what proteins will be bound to the operon in the different sugar conditions. So your protein options are RNA polymerase, the inhibitor, and the CAP activator protein. And then the conditions you should think about are when there is lactose, but glucose is absent, when there is glucose, but lactose is absent, when both sugars are available to your E. coli, and then when neither sugar is available. Another thing to think about is that you may have multiple regulators bound at the same time. So it is possible to have an inhibitor and an activator both present on the operon. In that case, RNA polymerase will not bind to the operon. So think back to what we said about how the repressor actually represses and why that may be true. This has to do with whether or not RNA polymerase can bind. So if the inhibitor is there, polymerase can't bind, even if there's an activator trying to recruit it to bind more often. So in this case, if you think of it kind of like a competition between the activator and the repressor in this case would win. Okay, I hope that was a good overview of regulation of the lac operon. Drawing this out for yourself and going through these different conditions will be very helpful for you as you are working to analyze your lab data and just studying for understanding of gene regulation. This is a great model because it has both an activator and a repressor, and it's kind of a straightforward pathway in terms of how those are regulated by our sugar conditions. Make sure you let us know if you have any questions.